Hi, everybody. It's Mrs. Mancuso, and really exciting. We are on to Unit 6. So we are on our last unit to get ready for the AP Bio exam. And you guys know I love this unit. This is all the DNA stuff. So let's get started with all of the experiments. So these are the experiments that led us to our understanding of DNA being the genetic information. So our first guy is Griffith. And he is the one that worked with the mice, and he had 2 different strains of bacteria. And one was the R strain, which stood for rough. And so if you think about it, it had all of these little like spikes on its edges. And so R strain bacteria did not kill the mice. But the S strain bacteria, S was for smooth, they had a special capsule around them. And so the immune cells couldn't really grab onto this bacteria. So it would end up killing the mice. And he had multiple controls, like he tried the R all by itself and it didn't kill the mice. And he tried the S all by itself and it did kill the mice. And then he heated up the R and it still didn't kill the mice. And the really unique one that was so strange is that he heated the S and put that with the living R, and it ended up killing the mice. So that's weird. That is not expected at all, because the R shouldn't kill the mice. And if you heated up the S, you're killing the S. So what the heck killed the mice? So his result is that he figured out that there must be a transforming factor. So a transforming factor was something that was being passed from the S to the R. Sorry, I don't know why it keeps doing those little black lines through my writing, but this transforming factor is something that is passing it on. And he noticed that with the heated S and the R, that the R cells ended up looking like this, which means they look like the smooth. So they were able to take in something from the heated S. And so what we now know because of this, so we call this idea transformation, based on his original term, transforming principle. So transformation is the fact that bacteria can release plasmids. So plasmids are small circular pieces of DNA that aren't the original single chromosome of the bacteria, but they're extra pieces of DNA. So they can release the plasmids and other bacteria can take them up. And then if another bacteria takes up that DNA and actually transcribes it and translates it, it will be able to make the proteins that the first kind of bacteria had. So if this is my S and I release this plasmid into the environment and the plasmid has the DNA to make the capsule, if I am an R bacteria and I take this in, even though I was an R, I can behave like an S because I have some of the S's DNA. All right, our next experiment, Avery, McCarty, and McLeod. So what they did was they took the all of the liquid from the environment, from the heated S and the R. So they treated this with all different enzymes. And basically, I want to treat it with an enzyme that is going to remove all the RNA, and another enzyme that is going to remove all the proteins, and another one that is going to remove all the sugars, and one that's going to remove all the liquids, and one that is going to remove all the DNA. So I can just put here another one, remove carbs, remove lipids. So run as many of these experiments as you can and see what happens. So when you do it, if the mice still die, so in this one, the mice still die, and in this one, the mice still die, and in this one, the mice still die, and in this one, the mice still die. what happens is I know the transforming principle is still there if you take out the lipids, and it's still there if you take out the proteins, and it's still there if you take out the RNA, and it's still there if you take out the carbs. So when you remove the DNA, all of a sudden the mice live. So that tells us the DNA is the transforming principle. It is the thing that is being passed on from the S to the R, because when you remove it, your experiment doesn't work like it used to. So the basic thing is, back then they were trying to figure out what is the genetic information. So the genetic information that gets passed on is the DNA, not proteins. At that time, they definitely believed that proteins were the important information, but all of these experiments are what helped lead them to understand that it was actually DNA, not proteins. All right, Hershey and Chase. So Hershey and Chase did, their experiment is often referred to as the blender experiment. What they did is they wanted to look at viruses. And the viruses they studied were called bacteriophages. So a phage tells me that it's a virus, and a bacteriophage tells me that it infects bacteria. So these are the ones that look like little spaceships like this. And so this is my virus, this is my bacteriophage, and here's my bacterial cell. And so they infect it by the process of taking and injecting their DNA into the cell. But Hershey and Chase said, okay, there's two parts of the virus. The viruses really only have two parts. So they have this protein coat, and they have either DNA or RNA in their center core. And so which one is going to be present in the bacterial cell that gets infected? So what they did was they used radioactive labels. And so if you want to radioactively label the protein and radioactively label the DNA and see what gets passed on. So now we got to think about, what element do you want to radioactively label to distinguish a protein from DNA? So proteins have carbon, but so does DNA. Proteins have hydrogen, but so does DNA. Proteins have oxygen, but so does DNA. Proteins have nitrogen, but so does DNA. So none of those would work. So they had to find something that was unique to proteins, and that is sulfur. So I'm going to put a little S like this. So I'm going to use the red color to show that they radioactively tagged the sulfur to see if protein was present. And the way that I think is easy to remember is that when you think about proteins and how they fold, they can form disulfide bridges, and that was something unique and cool about them. Whereas DNA RNA has the element phosphorus, and there's no phosphorus in proteins. So this was a unique way to look at it. So what they did is they ran two different experiments, and in the first one, they used this radioactive tag, and that labeled the protein. And when they looked at the bacteria afterwards, all of the red radioactive tags were in the environment. They were not in the cell. And then when they ran the experiment again, and they only used this radioactive label of the DNA, they found all of those radioactive labels inside the cell. So because this is what was being passed on, that lets us know that this is the genetic information. So again, this experiment was to figure out the genetic information is the DNA that's being passed on from a virus to a bacteria, and from really one bacteria to the next bacteria. All right. Our next, Rosalind Franklin, our next woman, she is the one that did all the X-ray crystallography. And X-ray crystallography allowed her to look at the DNA, and she kept seeing this repeating structure. It sort of looks like an X. And what it was leading her to the understanding of the double helix structure. Sadly, from doing all these X-ray tests, she ended up developing cancer, and she ended up dying. But Watson and Crick used her work to figure out the double helix structure of DNA. All right, Shar Goff. Shar Goff is the one I always refer to him as like going all around the farm and testing all of DNA samples from different organisms. And what he figured out was the percent of A was equal to the percent of T, and the percent of C was equal to the percent of G. And so he figured out the base pairing rules. He also figured out that these are different for each species. So each species has their own genome, their own set of letters, A's, T's, C's, and G's, but in different percentages. So if you have the same amount of A's and T's, those are the two that pair together, and the C's and G's pair together. And we now know about purines and pyrimidines, so when we get into structure, I'll make sure I mention that. Meselson and Stahl, they were studying DNA replication. And they had three basic ideas. So if this is my strand of DNA, I want to know when it replicates, does it do it as a 100% conservative process? So first of all, I'm going to put over here, these are all of my little nucleotides. So nucleotides, and I'm going to put radioactive tags. So all of these have heavy nitrogen. So that means they are radioactive, and I can see where they end up. So if you end up making a new strand that's all red, and you keep your template, the original blue, that is what we would call a conservative process. That is not what happened. Instead, this is what happened. One side stayed the same as the original template, and one side had the radioactive labels. And so that is how they came to figure out that this is a semi-conservative process. And the third one was the idea of a disruptive process. And that would sort of look like this, like all crazy, like some pieces get passed on from the original and some get passed on with the blue. And so this would be called disruptive. But that didn't happen. So that was a no, and that was a no. This is what did happen. So the replication of DNA is a semi-conservative process. All right, describe DNA structure. So DNA, we know, is definitely a double strand, and it forms a helix, which means it goes back and forth like that. And the two strands are anti-parallel to each other. That means they run in opposite directions. They are also complementary to each other. So let's take a little bit of time and talk about what all of that means. All right. On this side, the oxygen of the sugar. So first of all, this dark part right here, this is definitely my sugar. And if it's DNA, the sugar is deoxyribose. And so it's a 5-carbon sugar, and the oxygen is there, and you always count away from the branch. So the branch is right here. So carbon 1, carbon 2, carbon 3, carbon 4, and then carbon 5 is up here on the branch. So this side, the 5 from the sugar is higher than the 3' from the sugar. So this side here is running 5 to 3. On this side, the O of the sugar would be here. This is carbon 1, 2, 3, 4, and the 5th one is up here on the branch. And so this carbon, the 3 is above the 5. So this side is anti-parallel, which means it is running the opposite direction. It is running 3 to 5. In reality, I would actually draw this A upside down, this G upside down, and this C upside down, because they look upside down when they're anti-parallel to each other. All right. these little circles like this right here, this is my phosphate group, which is PO4. And in here, this and this, these are my nitrogenous bases. There are four kinds of nitrogenous bases, two types. So nitrogenous bases could be purines, which are the double rings. That is adenine and guanine. And then there's also pyrimidines. Those are the single rings. And those are the T's and the C's, thymine and cytosine. So you'll notice here's a purine, double ring. Here's a guanine, that's another purine, double ring. And then over here, a T and a C. These two are both pyrimidines, they're the single rings. And you always, every single run is the same distance, rung. Sorry, didn't come out very good. Each rung or step of the ladder is the same distance across. And that's because you always have a purine with a pyrimidine and a purine with a pyrimidine. Now this side has the purine on this side and the pyrimidine, but it always, always, always has that. Now, the next thing I want to point out, I need to do a little erasing so that you can see it more clearly. A's and T's only have two hydrogen bonds between them. These are my hydrogen bonds. Attractions, positive, negative attractions there. This between a C and a G has three hydrogen bonds. The implications for that are that it's going to be easier to break a strand that has a higher number of A's and T's than it would be to break a strand that has more C's and G's. If you have more C's and G's, you're going to need a higher temperature to break the additional hydrogen bonds that are present for each one. All right, what is DNA for? Relate DNA to chromosomes, genes, alleles, and traits. So first of all, DNA is the directions for building proteins. Proteins make up so much of your body. So proteins make up enzymes, they make up neurotransmitters, they make up channels, they make up muscle, they make up hair. There are so many molecules. They make up receptors. So things that have very unique shapes are almost always the proteins in our body. And so those are built according to the DNA code. So based on the A's, T's, C's, G's in order, that will determine the codon, and the codon will determine what amino acid comes in and how they are put together. So the primary structure, and then eventually how it will fold over and have its tertiary structure. All right, so a chromosome, so inside of one cell, you have one nucleus, and inside of here, if it is a human, you would have 46 chromosomes. So here's one chromosome, one strand of DNA. And you might be thinking to yourself, oh my gosh, Miriam, I thought they look like X's, like that. They only look like that after they have gone through S, and S is where DNA replication occurs. So after it, they don't look like single strands anymore. They are replicated in their sister chromatids. But anyways, this is one chromosome right here. Within this chromosome, I could have, like, I literally can't even make enough separations. Each one of these is going to be a gene. A gene is the exact sequence that is needed to build one protein. To build one protein. Now, you could have hundreds and even over 1,000 genes on one chromosome. So think about it. You only have 46 chromosomes, and 23 of them are like each other. 23 of them have the same types of genes. Now, the genes do not have to be the exact same code. Different codes for a gene are called alleles. And we refer to alleles as like dominant alleles or recessive alleles. These are the different forms of a gene. And those forms of a gene are different directions. One set of directions, let's say the capital A, will give you brown hair. And then another form of the gene, another unique set of directions, would give you blonde hair. So even though I know hair color is like polygenic due to multiple genes, that's like as much as you can simplify it, is that a form of a gene is an allele. It leads to a trait like hair color. That sequence, that specific spot is called a gene, and the gene is located on a chromosome, and the chromosome is in your nucleus. All right, moving on. When does DNA replication occur? So it occurs in interphase, and it occurs during S. So you will remember the interphase goes G1 and then S and then G2. So during the S portion of interphase, S stands for synthesis, and it means the synthesis of DNA, making a copy of all the DNA. All right, how is DNA replication regulated? So DNA replication only happens when a cell can pass a checkpoint. So you have to pass the G1 checkpoint for S to occur. And in order to pass the G1 checkpoint, you need high cyclin levels. Cyclins bind to CDKs. CYC, sorry, I missed a letter there. Cyclins bind to cyclin-dependent kinases. And so I remember last unit I was drawing, here's my cyclin and here is my CDK. And together, when they're like this, it is on. And so if you have enough of these cyclin CDKs that you can pass the checkpoint, so here is my cell cycle, G1S, G2 mitosis, and I'm going this direction. Here's my checkpoint. So in order for me to pass it and go this direction, I need to have enough cyclins bound to CDKs to pass that checkpoint. If I do, then I'm going to do S. I'm going to replicate my DNA. The way that you make cyclins is another signal transduction pathway. So there has to be a reason. There has to be a stimulus that is causing a cell to say, hey, we need to double up all of our DNA so that we can divide and each cell will get a set of the DNA. All right, moving on. Describe DNA replication and the enzymes involved using the following diagram. All right, so here is my 5'3" DNA. And so I want to look at it. Okay, so first of all, this is my replication fork. And pretty much our number one enzyme is helicase, because helicase is what breaks the hydrogen bonds and creates this replication fork. So here in the middle, this is going to be my helicase, because this is what's going through, like these are all A's, T's, C's, and G's, and this is what's breaking those hydrogen bonds. The next thing I want to do is I want to look at the direction of my new pieces. So over here, this blue is a template, and this blue is a template. But my new strands are the ones in red. This new strand that is made continuously towards the fork, this is my leading strand. And it's 5'3". 5'3" towards the fork is the direction that DNA polymerase works. So DNA polymerase is the enzyme that adds on the new nucleotides. So DNA polymerase. So it's adding on new unattached free nucleotides from the food that you ate. So A's, T's, C's, and G's. They could have come from a banana or a piece of bacon or a grapefruit, but they're all coming from things that were once alive. And basically right here, like if this is an A, DNA polymerase goes, okay, I need a T. And if this one's a C, DNA polymerase goes, okay, I need a G. And so it always attaches the appropriate corresponding nucleotide going in the direction from 5' to 3' towards the fork. Now, here's our DNA polymerase again over on this side. It built this piece first, then this piece second, and then this piece third. So it comes in, it has a little primer, it starts the replication, and again, it is building the piece 5' to 3' because that's the only way that DNA polymerase can work. But because it is building it in discontinuous fragments, these are called Okazaki fragments. And together, they make up the lagging strand. So the lagging strand, when you look at it, it's 3 to 5 towards the fork, but it was really built 5' to 3' away from the fork in discontinuous fragments. Now, because you have fragments, you need to connect those fragments. So this enzyme shown here in yellow, that's ligase, and ligase is like DNA glue. That is what is going to connect our fragments to each other to build one continuous side going away from the fork. All righty, there's 2 more enzymes that I just want to use in this little space because I didn't really have room for them. The first one's called topoisomerase, and this unravels the DNA ahead of the fork. So if I go back to my picture, my topoisomerase would be working way over here. So topoisomerase is helping to unravel it. And the next one is going to be along the sides here where the replication fork is forming. And so those are single-stranded binding proteins, and they hold the replication fork open. Basically, those bases would love to just go right back together. So the single-stranded binding proteins hold the fork open so that DNA polymerase can get in there and add the new nucleotides. All right, how do the directions within DNA get copied over so that they can be used to build specific proteins? All right, the directions within the DNA need to be made one gene at a time. Oh my goodness, I don't know where this black is coming from. Sorry. One gene at a time. Let me write that over again. It's a mess. All right, let's see here. One gene at a time into messenger RNA. And then that messenger RNA is going to go through some modifications, and it's going to leave the nucleus, and it is going to go to the ribosome. And with the help of rRNA, which makes up the ribosome, and tRNA, which brings amino acids to the ribosome, it will eventually make a protein. So in order to build a specific protein, we're going to go through 2 steps. We need to go through transcription, which is how we'll get our mRNA. And then we have to go through translation, which is how we're going to get our protein. All right, describe the process of transcription using the following diagram. So we have here, oh my gosh, that's so small, but that says initiation site. So there has to be a promoter, which has that TATA box, TATA. And so that is where a transcription factor binds to signal RNA polymerase. So when we were making DNA, we use DNA polymerase. When we want to make RNA, we use RNA polymerase. And you have to have a transcription factor because you have all these different genes on a single chromosome, and you got to figure out which one you need. And you got to figure out which chromosome you need. So transcription factors are very specific. Bind to initiation sites called promoters and start the process going. This molecule that is moving here, that is my RNA polymerase. And this that's being made right here, that's my messenger RNA. And so this was my original DNA. Here was my promoter, and this is going to be my terminator. All right, so you have to have a beginning and you have to have an end, and you basically read off the 5' your new 5' piece. So this piece that I'm building, this is the 5' end of it. This, the end, the part that it's growing, that's my 3' end. Here, I like this picture because it's blown up. It's like if I could take this thing and look at it really big, what would it look like? So the direction of transcription is going this way, which is making me my mRNA. This is going to be the 5' end. These new parts are going to be the 3' end. It is adding on free RNA nucleotides, again, from the food that you eat, and the breakdown of RNA after you've used it. You're going to have some of your own RNA as well. And so this is my RNA polymerase. And then the blue, this is my DNA. So if I'm making 5' to 3', this direction of the DNA template, so my template DNA is 3' to 5', because that will allow me to build a new piece that's 5 to 3. And when it hits this, the termination sequence, it lets go. And so here's my messenger RNA. It's not ready to leave yet, but that's the messenger RNA piece that I made. So the next question is, what are those modifications of RNA that have to be made before it can leave the nucleus? So the first step is that it needs a methylated, it's called a GTP. It is a 5' cap. And it does have guanine on it, and it does have 3 phosphates, guanine 3 phosphate. And you might remember that when we actually built our mRNA molecules from our DNA molecules. You had to take your big long chain that you made, and you added that single piece to it, and that was your methylated 5' cap. And then the second step is adding a poly A tail to the 3' end. So here's 5', here's 3'. You add on A. We only added like 6 of them to ours, but in reality, you could add like 100 A's to the end of it. And that is its protection on that end. And then the third step, really, really important, is called splicing. And splicing removes the introns. I always say introns are in the way, so you got to get them out. And this is done by a spliceosome or a SNRP. And so it is going to cut the piece. So here is my thing. And let's say here to here is an intron, and here to here is an intron. It's going to cut these parts out. And so then when it's put back together, these pieces that are kept are called exons. So the exons stay and the introns get cut out. So splicing removes those extra pieces. There's the cap, there's the tail. Now it's a little bit shorter, but it's ready to go. It has its modifications. How is transcription regulated? Distinguish between operons in prokaryotes and transcription factors in eukaryotes. So I'm actually going to do this one first. Transcription factors are specific proteins. They are usually the result of a signal transduction pathway, and they specifically turn on transcription in a eukaryotic cell. So if I have a eukaryotic cell, let me just take up a little bit of space here. Here's my receptor, here's my ligand. Here's my signal transduction pathway turning on kinases. And eventually inside the DNA, this process turns on a transcription factor. The transcription factor binds to a very specific gene on a very specific chromosome. That makes a piece of messenger RNA by the process of transcription, and that gets a cap and tail, and it gets spliced, and it leaves the nucleus through a little pore, and it goes to a ribosome, and it is eventually going to build a protein. So that is how transcription is regulated if your cell is a eukaryotic cell. if instead you have a prokaryotic cell, you're going to have an operon. So an operon is it's more than one gene. So I can't really call it a gene. It's a huge long piece of DNA. And remember, in a prokaryotic cell, this is a circular piece of DNA. So this is one part of the circle. So there is a regulatory gene here. Regulatory genes make repressor proteins. And they make the mRNA, and then the RNA goes to the ribosome, and the ribosome makes the repressor protein. So I'll make my repressor protein look like this, okay? And then the next section is the promoter. This is where my ta-ta box is. And the next is called the operator. And then you have your actual structural genes. Structural genes are what you need to make your mRNA, so that for this gene, you go to the ribosome and you build whatever you need. So let's say that this is the lac operon. The lac operon is inducible. Inducible. All right, inducible means it's normally off, but the presence of a substrate can turn it on. All right, so normally, normal conditions, this repressor protein right here, this would bind, sorry, it didn't keep the whole part with it. It would bind right here. And so if this piece is bound right here, this is going to block transcription. So what's it blocking? It's blocking polymerase. So right here, this would be my RNA polymerase. And if I take my pen again and I try and move it, this is stopping. It can't get any further. So right now, you would say that transcription is off. All right, we said that the way to turn it on was to add a substrate. So I'm going to start adding my substrate. And I'm drawing it as a double ring because lactose sugar, so this is lactose, is a disaccharide. And what happens is this, oh, I need to grab the little thing. Okay, this is going to move out of the way. And so if it moves out of the way and it goes over and it binds to the lactose that's now present instead, now my RNA polymerase can start working. And it's on and it's moving and it's doing its job. Yay. That means I'm going to make mRNA, and that means I'm going to make lactase. Not lactose, not the sugar, but I'm going to make lactase. Lactase is an enzyme that works on lactose. Now, as I make lactase, I will break down the lactose when there's no more lactose available. So I'll take my black pen here, I'm going to cross them out. I have now broken down all the lactose. When it has all been broken down, now this red repressor protein is going to go back and it's going to block the operator again. And when it blocks it, it's blocking transcription and it's back off. So this is a great example of how you could conserve energy. And this is what prokaryotic cells do. Why waste the amino acids to build lactase if you haven't actually eaten any lactose? You shouldn't. So regulation is all about feedback. When I need lactase, I should make lactase. I only need it when I actually have lactose present. All right, there is one more kind of operator that I need to show you. And so it is the trip operon. And that is a repressible operon. That means it's normally on, but too much product turns it off. Okay, so what happens with this one, here's my stretch of DNA. Here is my regulatory gene. This makes a repressor protein. The difference is the repressor protein that's made is inactive. So I'm going to draw it like this. This is my repressor protein. Okay, now this section is my promoter, this section is my operator, and this is my structural gene. This is my RNA polymerase. Since my repressor protein is inactive, it's going to stay down here. My RNA polymerase is going to be able to boop, 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 go right along. It doesn't have anything blocking it. Because it doesn't have anything blocking it, this is going to make messenger RNA. And that's going to go ahead and build a protein. And the protein is tryptophan. So I'm going to draw it as a little T with a circle. If I start building up too many of these T's with a circle, they will actually bind to the repressor protein, and they are now going to make it active. So if I have a repressor protein and the T all together, I'm grabbing too many parts. Let's say it's just this one little part, and this is both of these all active. This is now going to go up, and now it blocks. And so when it is active, so if the repressor protein is active, it is going to block transcription. And that was only when I had too much tryptophan present. And so no sense making tryptophan if you have way too much tryptophan. So again, it's another way of regulating when the gene should be on and when it should be off. All right, next question. All right, now we've got translation. So translation is using your mRNA and your rRNA and your tRNA to actually build a protein. The goal is to make proteins. So this is going to happen at a ribosome. So here's my ribosome right here. And B is showing my messenger RNA. And the direction that it's being read is 5' to 3'. That's why you see the 5 to 3. And then these molecules C, these are my tRNA. But these ones right here have already been used. So I got to back up a little bit. These ones are actually in the process of being used. So here is a tRNA molecule in its complete state, which means that it has E attached to it, and E is showing that this is my amino acid. So tRNA molecules stand for transfer RNA molecules. They bring the amino acids to the ribosome. And when they bring it, what's happening is this interaction right here. So this is codon, AAA, from my messenger RNA, interacting with the anticodon, which is UUU, from the tRNA molecule. So they pair up accordingly, and when they do that, the amino acids are really close together, and right here, they form a peptide bond. As they form a peptide bond, all of these are connected, so D is my growing polypeptide. And this process is just going to keep continuing, moving, boom, 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 until you hit a stop codon. And when you hit a stop codon, there's going to be a release factor, and it's going to let go. And then this growing polypeptide that I have here can fold over and make a unique shape. And that is going to be the protein that I built. All right, what is a mutation? So a mutation is the change in the DNA sequence. There are so many ways you could change a DNA sequence. First of all, you can have an overall change to an entire chromosome. So here is my big long chromosome, and let's say this little top half of it breaks off. That is a huge chromosome deletion. You are getting rid of an entire sequence. There might be 70 genes in that little section. This could be very devastating. They actually have figured out that a lot of miscarriages are probably due to drastic chromosome errors, drastic things that are not allowing an embryo to form. Smaller things that happen within just one gene could be something like a silent mutation or a missense mutation or a nonsense mutation. And so all of these are substitutions. So a substitution means you switched a letter. So let's say you switched an A for a U. And let's say you get the exact same amino acid. If you get the exact same amino acid, it's silent. If you changed an A to a U and it gave you a different amino acid, that's going to change your overall protein, and that's a missense. And a nonsense, let's say you change your A to U and now it codes for a stop. That's nonsense. That's crazy stuff. So that is, those are 3 types of substitutions. Another type beside a substitution could be an addition, which sometimes they call that an insertion. So it's like, let's say the original sequence was AUGCCACAU. Okay, those 3 letters, then those 3 letters, then those 3 letters. And insertion would be if I added a letter in. So let me just grab red, and all of a sudden right here I'm putting a G. Well, now it's going to read AUG, CCA, GCA. And so GCA is definitely going to give you a different amino acid than CAU. The earlier you have an insertion, or let's say I have a deletion, let's say I get rid of this U right here. Now it's going to read AGC, CAC, AU. That's also drastically changing what I code for, the amino acids that are going to come in order. And so when you change your sequence, you change your amino acid order, and that is going to build you a different protein, which could end up with a different shape. Chances are it's going to be a different shape. If a different shape, it's going to give you a different function. So one great example, sickle cell anemia. Sickle cell anemia, a normal red blood cell has a nice round shape like this, and it does like pillow in a little bit in the middle. But if you had a sickle cell, it has this really weird shape like this, and it has one amino acid change. Isn't that crazy? One change and it changes shape that much. So that's how you end up with sickle cell anemia, and it's simply due to one substitution in one gene. All right, let me just check and see. I think there was something else about this question. Oh, are all mutations negative? No, the fact that we have 5 fingers and that our 5th one is the thumb and that they're opposable, that is like an amazing mutation. Or having like really sharp eye teeth so that you're able to chew your food, or the fact that your vision is so amazing. There are tons of mutations that have been passed on generation to generation because they're beneficial. So there are definitely some mutations that you can think of that are bad, but there's also a lot of great ones in every single species that have allowed them to survive in their environment. So yes, they can definitely be positive as well. All right, determine the amino acid sequence for the piece of DNA below. All right, so if this is my DNA, and this is going to be 3' to 5', my new mRNA is 5 to 3, and it would go G. Now, I would want to write a T, but I can't because RNA doesn't have T's. It has U's. So GUG is the first one. And then CAC was the next one. And then CUG is the next one. And then ACU. And then GGG. and then UAA. All right. There's my codons. And those codons, I would have to go down to my chart to read what it would code for. So the first one, GUG. So GUG is giving me valine. And then the next one, CAC. So I'm looking for the first letter. There's the C. Here's the second letter, A. And here's the third letter, C. So CAC gives me HIS. And then the next one, CUG, C, then a U, then a G gives me leucine, LEU. And then ACU, threonine, THR. And then GGG, GGG is glycine. And UAA, UAA is a stop. All right, so that's my amino acid sequence. And after the original DNA sequence, alter the original, forming a mutation, what type of mutation you made and how the protein is different. So how would this change the overall function of the protein? So I am going to leave this for you to do. And so you go through, change the sequence. You could give me a silent, you can give me a missense, you can give me a nonsense, tell me how it changed it. And only if it's silent do I expect no change in the overall shape and function. If you do missense or nonsense, I'm definitely expecting a change in how it's going to look and then how it's going to behave. All right, what is meant by the term central dogma of biology? So this is that all cells have DNA. Every living cell has DNA. And then DNA goes through the process of transcription to make RNA. And then RNA goes to ribosomes in every single cell and goes through the process of translation to build proteins. So no matter if you are a turtle or a human, you have DNA, you make RNA through the process of transcription, and you make proteins through the process of translation. This tells that we are all united. We all have a common ancestor cell. And we all have the common unity and the central line of this is how every single cell works. Everybody's got DNA, everybody makes it into RNA, and everybody builds proteins according to the same set of directions. So once scientists understood this common thread of DNA transcription translation, they realized they could use it to improve species. So here we are as humans constantly trying to improve the planet. Not like, oh, I need to make this bug a better flying machine. No, we are thinking about how can we feed more people? How can we make the environment someplace where all different species can survive and have the chance to benefit from that? But if we can use species that we eat as an example, we are trying to improve crops. So to improve crops, we use recombinant DNA. So we are going to incorporate genes that are beneficial into plant foods. So let's just think about corn and apples for a second. These are two foods that people eat a lot of. So what would be beneficial? I want to have it be anti-drought, meaning that if there is a time when the crops are not getting rain, that they're not going to dry up and die. And antimicrobial, like I don't want bacteria to get inside of it and destroy it. And I want to have it have more vitamins or more minerals or more something that is going to be healthier and beneficial for you. So how will I get those genes out of the DNA? I need to use what are called restriction enzymes. So restriction enzymes cut DNA at specific sequences. The sequences are called palindromes. They're the same whether you go forward or or reverse. And so if this is my big, long, double helix DNA, the way that they work is they cut in, across, and down. And so what would result from this cut is I'm going to have two pieces. One is going to look like this. Together, 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 open, open, open. This was all together originally. All right, my other piece, here it is. Together, 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 open, open, open. These open parts are called sticky ends. So if I have two different samples of DNA, so I just did all this red and I cut it open. And here I have another one and it's blue and I go in with the exact same restriction enzyme. And so now I have this piece with its little open parts and this piece, and there's my sticky ends. I now have cut it with the exact same piece. So I should be able to take that and put this and this together. And so this is what I did with the necklaces in the classroom. Since I don't have room, I'm just going to go down here and draw it real quick. So let's say here is my starting red sequence. And then here's my other piece. Together, sticky ends, together, sticky ends. And then the blue pieces are going to seal right on. There you go. There's the open piece and the together. And now this piece is recombinant DNA. So I took and I added that beneficial gene into the original DNA. So let's say I took the drought resistant gene and it's the blue and it now fits right into the original corn DNA. That's how recombinant DNA works. And the enzyme that does the final sealing together is ligase because that is always the DNA glue. All right, I'm going to erase this because I really just didn't have enough room for it. And now I need to do this question number 18. So question 18 says, draw and describe the process of gel electrophoresis. Identify how it's used and what type of molecules can be separated in this way. So as I'm drawing it, I'm just going to talk about what it's used for. So it is used to separate DNA or RNA or proteins. We did it for DNA when we did it in class, but you really could use it for any of them. All right, DNA is negative, so you always load DNA on the negative side. These little parts you call wells. And down here, this is going to be the positive end. And so you are going to, number one, cut your DNA. You have to cut it before you load the gels. So you cut it with restriction enzymes. So here is my DNA, and I cut it into my little pieces, and then I load this into a well. So with this one, my restriction enzyme cut in 1, 2, 3 places. That gives me 1, 2, 3, 4 pieces. The piece that is the largest is going to be here. The piece that is the smallest travels the furthest. So the smallest fragment travels the furthest. And since DNA is negative, it moves towards the positive end. If I loaded another sample that was just like it, I would expect the exact same bands because it was the exact same sample of DNA and it was cut with the same pieces. If I was loading something different and I was just testing it, like, hey, are these really the same types of organisms? Like, you could use this to see, like, how closely related is this species to this species? Well, see how their DNA lines up for specific genes. If it lines up the same for many, many genes, it's showing similarity and that maybe they have a common ancestor. And if it shows a lot of differences, maybe they are not related and they are not from a common ancestor. If instead you were doing this with like a parent and a child, like a paternity test, let me erase these for a second. All right, let's say this is my child, and this is the mom, and this is who I think is the dad. The child's bands should all match up with mom and dad's bands. So if this one is from mom, maybe this band came from dad. And if this one came from dad, maybe the next one came from mom, like that. Half of the bands should be showing as coming from mom and half coming from dad. And that way you know that that is the father. So crime scene, paternity tests, seeing how closely related organisms are, and to see if you can detect a genetic disorder. Like if you're trying to find out like, hey, do I actually have the breast cancer gene? You could do a gel electrophoresis test to test for that. All right, next one. Since bacteria have no nucleus, they have unique ways of housing and sharing their DNA. Although they reproduce asexually, each of the following allows them to increase the variety in their DNA. Discuss each: transformation, transduction, conjugation, and transposons. Okay, so the first thing I want to talk about, prokaryotic cells. So prokaryotic cells, bacteria, have no nucleus. They have one chromosome, and that one chromosome is circular. So I'm going to show, this is my circular piece of DNA. It is not a nucleus, but it is a circular piece of DNA. And within it, you could have like one gene goes from here to here, and another gene here to here, and another gene here to here, and another gene there to there. It's small compared to human cells, but that is their circular piece of DNA. But then they have extra pieces of DNA. So these extra pieces of DNA are called plasmids. And so we know that this is definitely where antibiotic resistance genes reside. And probably a lot more genes reside there as well. But they are genes that cells can undergo transformation. So that first word, transformation, means that you can let out, not your circular chromosome, but you can let out your plasmids. So transformation. Here, this cell let out this plasmid, and then another cell could take in this plasmid. And now I have the genes of that cell's DNA. I have its DNA. So transformation is a way to increase variety because it is literally passing on DNA from one cell to another through the environment. Pass on plasmid DNA through the environment. All right, so this guy let it out, this guy picked it up. That is how they pass on antibiotic resistance, and that is called transformation. And we did it in lab, and you can do it with a lot more things besides resistance. We just happen to do that. All right, the next word, transduction. Transduction is what a virus does. So a virus, like a virus that would live on a bacteria, looks like that special little spaceship look. And so it will inject its DNA into this cell. So now this cell can incorporate the viral DNA. Incorporates viral DNA into its genome. So your genome is all of your DNA. So now this little piece becomes a part of, like if this is my circular piece of DNA, it'll just take it in. And now it's part of the circular DNA, which means it is going to build these guys. It's going to be a factory for making viruses. And so that's the problem right now. Like if that's the coronavirus is a retrovirus. It has RNA. It injects its RNA into cells. Cells replicate the RNA. They make more copies of the virus, and that is how it spreads throughout your body. And when you cough, it is in the droplets and can go to another person. All right, that is definitely increasing variety because you incorporated new DNA into your bacterial cell. All right, so we did transformation, we did transduction. Okay, conjugation. Conjugation is direct contact DNA exchange. So here's one bacteria, here's another bacteria. Sometimes they have these little things called pili, which are little like extensions off of them, and that's how they connect to another cell. And if they pass, like here's some DNA inside of this cell, if they pass it from one cell to another cell, they are definitely going to increase the variety. But the difference is this one, it was in direct contact. It wasn't through the environment. And the last one, transposons. All right, transposons are what they call jumping genes. And so these were especially studied in corn, and they saw that genes actually can move from one bacterial cell to another bacterial cell, and can spread very quickly. So these little jumping genes just moved from cell to cell. Again, these words don't necessarily come up so much in AP Bio, but just wanted to expose you to all of them because they do come up in biology over time. All right, next page. In class, we did the transformation lab, and we were able to make E. coli be both ampicillin resistant and glow in UV light. Explain how we did this and what negative controls we put in place. Okay, so we know that if this is my E. coli, that they grow in what are called colonies. And as a colony spreads, it sort of takes over the whole plate, and it can develop what's called a lawn. And on this Petri dish, you need things to keep it alive. So you need luria broth. That's basically food for bacteria, and it needs to be incubated. Usually we keep it around 37 degrees Celsius, a nice warm temperature that's a lot like human body temperature. All right, so what we did, we took our E. coli, and so picture this as a little bacteria cell, and we gave it a plasmid in its environment. So all of these are our plasmid, and our plasmid had two genes. It had the gene for ampicillin resistance, and it had the gene P-glow to allow it to glow as long as the presence of a certain arabinose sugar is present. All right, now these E. coli, they could just let this in, but we actually did something to them. We heat shocked them. And when you heat shock it, oh, I'm going to take this in. I'm going to take this in. It increases the permeability of the membrane, and it increases the chances that the E. coli will take it in. So when you set up your plates, I could set up a plate with luria broth, and I could have a negative. And a negative means no plasmid. If there's no plasmid, this is a great example of a negative control that we did. So we wanted to show through this plate that bacteria will grow. So we ended up seeing a lawn. So this was proof that our E. coli could grow under our conditions. All right, we had a great incubator, we had a great setup, we had great luria broth. It was great. You can't go back and say, you know what, our incubator wasn't good. This was proof that we had a good situation, and that's what you use a negative control for. But then we want to set up more tests than that. So we have one with luria broth, Anamines, and ampicillin. So ampicillin is an antibiotic. An antibiotic kills E. coli. This is another great example of a negative control because we are expecting to see no growth. And so the proof of this is that ampicillin works. Ampicillin kills E. coli. And we know that it kills E. coli because we ran this test to prove that it kills E. coli. No growth means ampicillin worked, because the only difference between these two is the presence of ampicillin. All right, then we want to show our pluses. So here's Luria broth plus. Plus means that it got the plasmid. And again, it was exposed to the plasmid. We cannot make it take in the plasmid, but we exposed our E. coli to the plasmid. And so if they got the plasmid, we expect to see growth. And we did not see a lawn, we saw colonies. And these colonies are proof that they took in the plasmid. And the reason why we know they took in the plasmid is because now, oh, sorry, I forgot to put ampicillin there. They are ampicillin resistant. Ampicillin resistant. All right, so these are 100% resistant. They would not be able to grow there if they weren't resistant. So that is why we did it. And then we had another one with Luria broth and the plus and ampicillin, but this one we also put arabinose sugar. And arabinose sugar turns on the P-glow gene. And so in this one, again, we saw our colonies, and our colonies are 100% transformed. So they took in the plasmid. And now the unique thing is that all of these are glowing. Glowing, 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 glowing. When you shine UV light on them, they glow. And that's because they have the inducer molecule. When the inducer molecule is on, the P-glow gene is turned on and it makes the glowing protein. So that is what we did, and those are the controls that we set up for it. All right, next slide. All right, describe each. So cloning is when you take and you remove the nucleus from an egg. And so we're going to call this mom number 1. And we take another cell. So we take a somatic, which is a body cell, which is diploid, from mom number 2. And we take it out. I already wrote that. Take it out and put in mom number 1's egg. All right, so here's mom number 1's cell. And so this was an egg cell, and it used to have N chromosomes, and now they're out. They're taken out. And here is mom 2's cell. And here is the diploid nucleus, 2N, and this is taken out and it's put inside of here. So now the diploid nucleus is inside mom 1's cell. And then mom 1 has a baby, and the baby is a clone of mom number 2. All right, so that's how cloning works. All right, PCR is just a process to amplify a gene. So amplify means make many copies of a gene. So let's say at the U of R, they're studying the breast cancer gene. Before they study it, they're going to want to make replicates of that gene over and over and over again so that they can do many, many tests. So that is what it is. And RFLP, that stands for restriction, fragment, length, polymorphism. And so RFLP. That means from me to you, sometimes it's pretty difficult to tell our DNA apart. We have so many of the same genes. So in order to do in order to do that, so this is to tell individuals apart, especially in like crime scene scenarios. You have to look for polymorphisms. You have to look for areas in their DNA where there are differences. Poly means many, morph means different or shape. So we want to find areas of many differences. All right, DNA sequencing is just figuring out the order of the bases for a genome. So like the Human Genome Project figured out all the A's, T's, C's, and G's in all of our DNA, all of our chromosomes. All right, and my last question relating to COVID, what is a retrovirus and how does it use its host? So a retrovirus has RNA, not DNA. So COVID is an RNA virus, a retrovirus. And so how does it use its host? So it inserts its RNA into the host cell, and then the host converts the RNA into DNA. And so that is the reverse of transcription. So the enzyme that does this is called reverse transcriptase. And the DNA that's made is called cDNA. And the C stands for complement or complementary DNA. It is literally what people call perfect DNA because it has no introns. So it's a very effective virus because you don't have to, as a cell, as a virus, you want to take over your host and have your host make your DNA. If the host has to figure out which parts are the introns, it's never going to do it right. If there's no introns in it, it's perfect. So another virus, the HIV virus, is also a retrovirus. So a retrovirus is able to easily take over its host cells because it goes in and it changes its RNA back into DNA, and then the cell will just use the DNA and make the proteins that the virus wants it to make. And eventually, this host cell is going to make all of these. So like picture on the news, these are all coronaviruses. It just did that until eventually this cell breaks and it releases all these coronaviruses into other parts of the body, or every time you sneeze, you are sneezing out that virus. You are making copies of the virus. So the host is making copies of the virus. That's how a virus works in your body. And I really wanted to ask a question too about antibiotics. Maybe it's on this next page. Oh yeah. What do humans use to control bacteria versus virus infections? So when you have a bacterial infection, you take an antibiotic. An antibiotic is a medicine, and that medicine attacks the coat of the bacteria. If you have a virus, there are no antibiotics for viruses. And that's usually why you just live out a cold. Like you have a cold is a virus, and your immune cell works. And so your immune cell is going to work for both of these. Immune cells. So your immune cells are going to make antibodies. Antibodies are Y-shaped proteins that will attach to cells and will destroy them. Now, it doesn't matter if this is a virus or a bacteria. Anything that's a pathogen can be attacked by an antibody. So when they say that they want to test people and see if you have the antibody for COVID-19, this is going to be very specific for COVID-19. So they're going to be able to say, oh, you have this exact one. You made that because you had COVID. If you didn't have COVID, you don't have it. You won't find it because there isn't any present because you didn't make it. You only make the antibodies for things that you're fighting. So like you can go back through and you'll have the antibodies for all different things that you have had before because you have what are called memory B cells. And memory B cells in your immune system, they remember all the different antibodies that you've made in the past. And so when they say, oh, you never get sick with the same thing twice, that's because you have the antibody for it. So if you have this memory B cell and all of a sudden here's COVID-19 and you've already had COVID-19, this cell is like, oh, here we go. I'm making 1,000 antibodies, and all these antibodies are specific to COVID and they all kill COVID. So that's why you're not going to get it a second time. And now when they say like, oh, but COVID, they might end up finding out that you can get it again. That's because if this is the original COVID and this is the new COVID, it's taking on a different shape or it has different receptors on it. The name COVID or the name coronavirus actually, so CO stands for corona. Coronavirus is because when you look at it, it looks like all these little crowns on it. That's how it got its name. Corona means crown. And so if those change, then the antibodies need to change. And so if you get it again, so you can think about this like a cold. You have gotten a cold more than once in your life, and that is because the virus changed. If COVID-19 changes, you will be able to get it again. Do scientists know for sure if it will or won't change? Nope. The only thing we know is that time will tell. Now, the other thing in the news, vaccines. So when you're getting a vaccine, here's that little coronavirus. You are getting either a dead or a weak form of this into your body. So here's happy little you. They are injecting, like let's say they're injecting it into your arm, or let's say it's one of the ones that you take like an inhaler and you breathe it in. You are taking in this dead or weakened COVID. And now your body's immune cells, your B cells, are going to work until they make the right antibody. And when they make the right antibody, they are going to kill the coronavirus. And then you are going to make memory B cells so that if you do come in contact with the active COVID, not the weak one, but the strong one, your body's right away going to be like, I'm not going to get sick from that because I have memory B cells and I know how to make an army of antibodies really, really quick. So that's how it works. So I know this was a super long one, but hopefully it was really helpful. And that was our last unit review. So go through, see what questions you have. Don't forget that we Zoom every single Thursday at 1. I go through all the questions from the AP classroom where there's only 2 of them, but I go through anything from this and then any questions that you have from the AP classroom. All right, miss you guys.